question, I like indoctrinate, like coincidentalizing, just because it's magic and it's classy, so that's all we learn about.
All right, let's go ahead and get started here. The first thing I want to bring to your attention, I'm giving you yet another survey, but this is for my own information. I feel like the videos I've been assigning are great, but maybe it's turning into busy work. And I don't want it to just be busy work. I want it to mean something. So I'm thinking of maybe switching from not doing these video quizzes to doing maybe some other type of homework. It would be yet another like short, fun type of homework assignment. And I know it's a change in the schedule, so I want to get your all opinions on it so that I am not, changing things unnecessarily. Or maybe you really like the video quizzes and I should keep with them, but I want your opinion. So if you could take this survey, it shouldn't take too long here before we get started. And we are going to do the rest of the content check today as well. I have an alarm set on my watch, so we have enough time at the end of class to finish that up and turn it in. So if I'm still in the middle of a lecture when my alarm goes off, I will finish it up at that point. Alright, if you're still filling out that form, that's fine. I'm going to keep us moving here. Because today we're going to talk about our last two biomolecules. And we're going to start with proteins, alright? Proteins, super, super important. In our body, they are prominent in pretty much every body function that you can think of, right? They can be enzymes. They can act in defense. They can help move things within our cells. They can signal between cells. They provide the structure of our cells themselves, as well as transport many different materials. They are super prominent. We will talk about very specific proteins as we move into things such as photosynthesis and cellular respiration. All right. I just want to throw a whole list up there to show you that, yes, proteins are important. All right. So proteins, they are involved in most bodily functions. And they are very, very diverse. All right. There are thousands upon thousands of proteins. And there are so many because a singular protein has a very selective function, meaning it will likely only function in one specific thing. Right. Now, there can be some cases where a protein might do a handful of things. It's more rare, but typically it's a protein to its function, right? And because they're so specific to what they do, the structure of this protein is really going to dictate how it functions, right? And you will see structure equals function many times on these slides because it is so crucial. Now, the interesting thing about proteins is there might be thousands of them, but they're only made up from 20 amino acids. So 20 amino acids is the entire building block for all of these proteins. I put up just kind of a nice little diagram of all these amino acids up here, and we see that amino acids, they can have different properties, right? But there's just 20 of them that make up every single protein in our body. Once again, my clicker's not working today. Try that. All right, so the general structure of the amino acid, right? All amino acids are going to have this general structure to them. They're going to have an amino group, right? So it's highlighted in pink here. It's going to be positively charged. Right? They are going to have a carboxyl group. So again, this amino group and the carboxyl group, those are those functional groups we were talking about on Wednesday. This carboxyl group is going to have a negative charge associated with it. There's always going to be a central carbon, right? That can be called the alpha carbon. And off of that alpha carbon, we are going to have a single hydrogen atom. Now it says R group, that's highlighted in blue here, that makes every amino acid different from one another. So I'm just going to briefly go back to that previous slide here. And those R groups are all highlighted in this chart. So they all have an amino group, they all have a carboxyl group, the central carbon, and that singular hydrogen. But then they all have this R group that changes slightly between them all, right? And it's that R group that gives them kind of like how they're going to structure the rest of the protein, as well as their functions. Right? So I may not ask you to draw this, but if I give you a drawing of an amino acid, you should be able to tell me, yeah, that's the amino group, that's the carboxyl group, that's where the R group, hydrogen, and carbon would go. Right? Because it's that important of a molecule. Now, so you can go back to this slide here. So these are the monomers of our proteins. Right? We're back to thinking about the monomers that will combine together to build those polymers. Amino acids are the monomers, right? And when they combine together, right, they will form polypeptides. That is the polymer. Right? So a polypeptide is a chain of amino acids. Right? It's a simple chain. They're going to be joined together via peptide bonds, hence the name polypeptide. Now the peptide bond, it's a covalent bond. It's just a specific name for the covalent bond that binds two amino acids together. And as we have seen multiple times now, and will continue to see, they bond together via a dehydration reaction. Right? So we have OH group and a hydrogen group. They're going to combine to form water. Water is going to leave this reaction, and what remains is this peptide bond in its place. Right. I threw up this video because I like visuals, and I think visuals of polymers forming are very helpful.
During protein synthesis, peptide bonds link amino acids together in the order specified by DNA instructions. In this case, the first two amino acids in the protein are methionine and alanine. Here are ball and stick models of these amino acids. Peptide bond formation is a type of condensation reaction. During the reaction, the highlighted atoms are removed. The carbon atom in the carboxyl group of the methionine becomes linked to the nitrogen atom in the amino group of alanine. A peptide bond forms between methionine and alanine. Water forms as a byproduct of the reaction. The next amino acid specified by the DNA is leucine. Again, atoms are removed and a new covalent bond forms. Another molecule of water forms as a byproduct. The next amino acid is tryptophan. Another peptide bond forms and another molecule of water is released.
All right, so I like this video. It's simple, it's to the point, it gives you a really good visual. But again, dehydration reactions, they call it a condensation reaction, same thing. All right, so that's a polypeptide. Right? Polypeptide is a component of a protein. So a protein is one or more polypeptides that combine together in a really complex three-dimensional shape. Right? So polypeptides that have been twisted and folded and forming these complex structures that make up a protein. Right? So if I call it a polypeptide, right, it could be a protein. Right? Protein can be one polypeptide. It could be a singular part of a protein, and you need more polypeptides to join together to make up the final product. Now, something I just wanted to note and throw out there is that these proteins can have they will have amino acids joined together, but they can have these carbohydrates and lipids attached to give a unique functions to those proteins. We're not really going to talk about that, but I want you to be aware that other attachments can occur. All right, so let's talk about protein structure. right? So protein, it's not just a string of amino acids, it's the way it's folded into this three-dimensional shape. And there's multiple levels of structure when we talk about a protein, right? All the structures are up on here, and we're going to go through them one by one. But I wanted to start with the primary sequence. It's easiest to understand. It's what we have been talking about so far. It's simply a chain of amino acids, right? There's no folding occurring. It's just a simple chain of amino acids. Okay. Now, it starts to get more complex when we think about the secondary structure, right? So this is where we start to have some chemical interactions between our amino acids, right? And we get protein folding, right? The very basic level of protein folding. Now, in the secondary structure, we might have these specific formations that we would call alpha helixes or beta pleated sheets. Right? So I have this helix up top and the sheets down at the bottom. The sheets kind of, if you think if you folded a piece of paper like accordion style, right? Now, what holds these together are going to be hydrogen bonds. And hydrogen bonds are super key here. The same image, but with our molecules overlaid on top of it. All these dotted lines here indicate a hydrogen bond, and we can see for this secondary structure, this hydrogen bonding between amino acids within the same polypeptide chain is really what's giving it this folded structure. Right? So hydrogen bonds are key. Now we can have other types of structures in our secondary structures of proteins here. They're coined these random coiled regions, right? I'm sure some of them might have names, but for the basics of 101, we're calling them random coiled structures because they're just any structure that's not a helix and a beta pleated sheet, but they still have very important functions because of the shape that they are in. Right? If we go up yet another level of folding, we would call this the tertiary structure. Right? And so the tertiary structure is folding these proteins even more, giving an even more complex pleating shape beyond just helices and beta pleated sheets. Right? Now, I have not gone into depth about how this happens, but I want you to understand that it's the charges on those R groups that really play a crucial role here. Right? We can have hydrogen bonding, giving these three-dimensional shapes to our protein. We can also have ionic bonding and covalent bonding. We can have interactions between specific atoms. Right? There's something called a disulfide bond that can occur that also helps create this structure. Right? But really, it's these charges. And we can see here that we have nonpolar, we have polar, negative charged, and positively charged amino acids. Right? How they all kind of interact with each other determines on which sequence they were initially built into that polypeptide chain. All right, so this is kind of like our third level of structure here, and it's the last level of structure for a single polypeptide chain. Right? So for a singular chain of amino acids, this is as far as it will go to being folded. The proteins can get more complex, all right? And this is the last level of structure. It's the quaternary structure here. And this is where we have proteins that are made up of more than one polypeptide. Right? So we have more than one polypeptide that's gone all through all these levels of folding. And now these polypeptides that are all nice and folded in these complex 3D arrangements are going to join to one another. Right? And when we have proteins that have multiple polypeptides, we would call each polypeptide a subunit. Now I think hemoglobin here is a really good example of this, right? It's made of 4 subunits, and each of these are color-coded in the diagram here. For hemoglobin, it just so happens that they are the same polypeptide bonded together into this shape here, right? But we can have proteins that have different polypeptides bonded together, right? So that we would call a multimeric, I should say, multimeric protein. And the types of polypeptides are different. Right? There's a lot going on here. Right? There's a lot of folding. There's a lot of different structure. different charges are interacting with other charges on opposite amino acids, all right? You could spend a whole semester talking about structures of proteins. In fact, we have a proteins class dedicated just to proteins here at Gustavus, all right? So I want us to just kind of step back and think about all this folding, all right? I want us to get into those groups that we have been working with here. If your group member is not here today, if you could join with one of the groups on your sides, that would be awesome. But I want you to imagine that a protein's primary structure could be altered. Right? Would this affect its tertiary structure? And even beyond that, would that affect its quaternary structure? So I am giving us 4 minutes, which might be a little long, but it also gives you an opportunity to ask me questions if you have them. So Ethan, I don't know if you want to join this group over here or over here. Maybe I want to go over here by yourself. Okay. Yes, oh, okay. Yes. be awesome to get over drink them. All right, so that is 4 minutes here. So what do we think? If we change a primary structure, is it going to affect the tertiary structure? Yes. How?
Okay, so basically in the primary structure, your process called transcription and translation, there might be like a whole different amino acid that is added in what it should be. So then by the time it gets to folded, it's going to be in a different shape, which could either be a silent mutation, so it can really not affect you, or it could be really detrimental and affect you.
Yeah, it's going to determine its shape, right? We went into a little bit more depth, and we won't actually be talking about some of those topics until our 3rd unit. But that's great. That's awesome. Yes. Do we have anything else to add?
Like specifically in the way that it's into shape, like so the argument typing now. So if it's meant to be, because it's part of it, right? It's meant to be polar and then to be non-polar, it will attract the arguments of different amino acids. It's changing stuff.
Yeah. Yeah. We'll have interactions between different amino groups than what we have originally been intended. Yeah. So think about, swapping a polar for a non-polar, or if you swapped an amino acid that had a positive charge, maybe it originally had a connection to a negatively charged amino acid, but if we swap it to a negative amino acid, negative to negative, they're going to be repulsed. And it's going to change the ultimate shape. Yeah. Do we have any questions on this? Right. Lab next week is going to kind of build upon this topic. Specifically about enzymes, which we probably won't get to by the time we have lab. But this will be some good foundational knowledge to get you through. Alright, I put sickle cell disease up there because it's a great example. And it's an example we used in lab this week. If you change one amino acid, it can have detrimental effects, and then it's going to change the entire shape of a red blood cell. Alrighty. We're going to keep cruising through then. So, again, structure equals function. Alright? So, proteins, they serve all these different functions in our bodies. And it's really the structure that's going to determine what function that it does. Alright, so now, we can have, if we have like a mutation and we switch out our amino acid in its primary structure, that can change its shape, right? But we can have other environmental effects that could also change the shape of a protein. Specifically, we could denature the protein. Right? So if we denature a protein, we're completely losing its shape. Right? A lot of those bonds that we had initially are going to fail. pH can denature a protein, whether it's too basic or too acidic. If it's outside of the range that that protein was originally designed to be in, we can denature our protein. Salt can denature our proteins. If we have too salty of a solution, for example, it can start to ruin some of our folding processes. And temperature, right? Oftentimes it's the hotter temperature that's going to denature it more than, say, a colder temperature. Yeah.
Does this happen in organisms in general, or is this kind of
Cool. So this could happen in organisms in general. So if you think of your body, we highly regulate our bodies, we keep it in homeostasis. What if that homeostasis gets out of whack? Right? We could denature some proteins, and depending on the extent of that, we could have some major consequences. Yeah. Same with other organisms out there. Definitely. Does that answer your question? So, I'll have a good example of what. If some pH gets out of whack in your body, it's going to have consequences on your things. All right. Just asked if there are questions, but if there's any more. All right, we're going to move on to the last biomolecule that we're going to talk about, and that is nucleic acids. Now, when we talk about nucleic acids, we're really talking about like the backbone of DNA and RNA. Now we're going to get much more specific into the processes of DNA and RNA and transcription and translation in the third unit here. But for now, we're just talking about the biomolecules that make them up. Right? So our monomers in this case are nucleotides. Right? Now nucleotides, they provide the backbone for both DNA and RNA. DNA is going to be double-stranded. It's going to stay in the nucleus, and it stores our genetic information. RNA, very similar, except now that it's single-stranded, right, it has the ability to leave at the nucleus, and eventually it's going to encode different proteins in our body. Okay? It's not going to turn into a protein, but it provides the information to make a protein. All right, just like we talked about the structure of an amino acid, we're going to talk about the structure of a nucleotide. So a nucleotide has three basic components. Again, if I gave you this image without the labels, I would expect you to be able to label the molecule, but not draw the molecule. Okay? So we have a phosphate group, and the phosphate group is up in yellow here. Okay? We have this 5-carbon sugar. Okay? It's always going to be 5-carbon sugar, but depending on if we're talking about DNA or RNA, that sugar is going to vary. It's either going to be ribose or deoxyribose. And then we have a base. Okay? A base is going to be at least 1 ring of carbon, but it can be 2 rings of carbon and nitrogen. So in this case, this is a 2-ringed base. This is adenine. Okay? So we have those 3 basic properties. We have the phosphate group, we have the sugar, we have the base. Now, I really like this image because it kind of breaks things down into the structural components. So if we're starting with our nucleotide here, okay, we're always going to have this phosphate group. No matter if it's RNA or DNA, always going to have the phosphate group. The 5-carbon sugar here, it can either be ribose or deoxyribose. Right? The only difference between the two is the addition of this oxygen in RNA. Right? So this one atom is changing the entire property of our sugar here. If we think about all the different nitrogenous bases here, you can have a whole series of bases. Right? Some of them only have that 1 ring. Right? When they only have the 1 ring, we call them primitives, and we will expand on this later. Right? If they have 2 rings, they are purines. In DNA, we will have cytosine, thymine, guanine, and adenine. Now in RNA, besides that different type of sugar, we would also have uracil in replace of a replacement of thymine. And then we'll talk about why. Yeah, I was going to say a few weeks, but it might be a couple months. Right? That's all the way in Unit 3. I just want you to know RNA can have that different use. All right, now if we're talking about how we're going to build our polymer, okay, so we have all of our monomers, we have our nucleotides, they're going to be combined together to form what we call a sugar phosphate backbone. And so if you look in this image here, this is one side of DNA, so this is one DNA strand. In the red is our sugar phosphate backbone, and it contains exactly what it is. Right? The sugar phosphate backbone is going to contain sugars and phosphates. So you can see we have a sugar, and a phosphate, and then a sugar, and a phosphate, and a sugar, and a phosphate, and so on and so forth. Now, when we get into this a little bit more in depth, we'll start to talk about the 5' and the 3' carbons. Right? For this part of Bio 101, I just want you to show how they play into making that backbone structure. So if we have our sugar here in DNA, you see that they're labeled 1, 2, 3, 4, and 5. Right? So I would say this is a 1' carbon, 2', 3', 4', and 5'. It's this 5' and 3' that are going to link to a phosphate group and build the entire chain. So if you look over here, we have our 3' carbon that's linked to our phosphate, which is then linked to the 5' carbon of the other sugar. Right? And it kind of builds in the same aspect going all the way down. Now, you could probably guess it's a dehydration reaction that makes this happen, right? So again, we are taking the OH from the phosphates and the hydrogen from our sugar here. We're combining them together to create a water molecule that will lead the reaction and leave behind a bond in its place. And again, we have a specific name for this bond. It would be a phosphodiester linkage. It's a covalent bond, but that's a specific name for the covalent bond between nucleotides. Now, you probably know DNA has this double helix structure, right? And I just want to point out for right now that this double helix structure is in part due to these hydrogen bonds between our nitrogenous bases, right? So in our nitrogenous bases, adenine is always going to link to thymine, right? Guanine is always going to link to cytosine, right? But if we look at those linkages, from adenine to thymine, we have 2 hydrogen bonds, and from guanine to cytosine, we have 3. Right? So they're going to play a role in twisting up this strand of DNA. And my alarm on my watch is going to go off in like 30 seconds. So that was absolutely perfect timing. Okay? That's where I'm going to end content today. I am going to say the homework for Monday, I'm not going to assign anything other than just remember to do your quiz for lab. And keep in mind that the exam is upcoming. Next week, I'm hoping to get at least study that. Right? So I am working on it. I am in the process. All right, let's do our content check. So at this point, you've done most of it, but you are missing the questions for part 2. Okay? And so part 2 questions, it's going to be a little bit more intense just because you have each other to work with and you have me to ask questions. I want you to take 15 minutes and finish it up. And once you're done, you can hand it in to me and you are good to go. I will say these content checks, there are 10 of them throughout the semester. Your 3 lowest grades will be dropped, right? So there's a bit of leniency there. All right, so basically for this first one, I'm going to explain it to you a little bit here. I want you to draw kind of like within the Bohr model of an electron, where we have our nucleus and then we have our atom, and then our atoms or electrons encircling the nucleus here. I want you to draw that arrangement of our atom, but I want you to draw that arrangement as if they were bonded together. So we have a nitrogen and 3 hydrogens that are going to be bonded together, and I want you to draw me that molecule, showing what electrons are being created. All right. I would start by drawing nitrogen and then go from there. And do feel free to work together on this question.